Good afternoon, everyone. I am Dr. Mohit Chaudhary. This is TransConnect February 2026, and this time we are going to talk about a very important topic in histocompatibility testing, that is complement-dependent cytotoxicity crossmatch, or the CDC crossmatch, or micro-lymphocytotoxicity test, which is a cell-based immunological assay to detect HLA antibodies that are donor-specific. That is a donor-specific antibodies. It is a very specific test but highly insensitive, and there are ways to actually make it more sensitive. The various methodologies in this, or the various modifications, are if we can extend the incubation time, when we increase the temperature, increase the washing test, and most importantly use the anti-human globulin or AHG with it. So this test, it has got its own perils. It's a very exhaustive test, but then it has got its own advantages, because this is like an in vitro transplant or a mini transplant outside the body, one. And secondly, because you can actually differentiate between an auto crossmatch and an allo crossmatch in this, so which is equally important. We know that auto antibodies are not deterrent to the graft, so even if you have an auto antibodies, you can still go ahead with the transplant. So that is another part of it. Other important thing is you can do a titered crossmatch also to look for a prozone phenomena, or report the titer itself to see at what titer it is giving the highest positivity. So that will actually help us to know when you repeat the crossmatch next time as to if the antibodies are decreasing or it is at the same level. So this CDC crossmatch gives you a lot of information. It tells you the complement acting antibodies, and also because it is a gold standard or a mandatory test before we proceed for the transplant. So over to Ms. Akanksha, who is our colleague, who is going to be talking about CDC crossmatch. Happy learning, everyone. And if there are any queries regarding this topic, you can always write on the chat box. Thank you so much. Over to Ms. Akanksha.
Hi everyone, a very good morning. Myself Akansha, and I'm from the Department of Molecular Biology and Transplant Immunology. Today we'll talk about a cell-based immunological assay, a silent test that predicts whether the graft will survive or be rejected within the hours of transplant. That is CDC crossmatch. CDC stands for complement dependent cytotoxicity, an immunological assay that was introduced by Patel and Terasaki in 1969. It is a classical assay used to detect the donor-specific antibodies. But why do we need to detect these donor-specific antibodies? What is the need? We need to detect these donor-specific antibodies in the recipient serum only, as if there will be any DSAs present in the recipient serum, they might be non-HLA or anti-HLA. But due to the presence of these antibodies, they might be donor specific or non-donor specific. Due to their presence, it can cause the hyperacute or the acute rejection in the patient, and eventually the patient life will be at stake. And our main and the prime responsibility is to ensure the patient's life. That is why we need to detect the donor specific antibodies. It is based on complement dependent cytotoxicity. It prevents the hyperacute rejection by the detection of donor specific anti-HLA antibodies in the recipient serum. It is a gold standard for pre-transplant compatibility testing. Here comes the principle of the CDC crossmatch. But first, we need to know what is HLA. HLA stands for human leukocyte antigen. It is highly polymorphic in nature and it is present on the chromosome number 6, basically on the short P arm. It is mainly responsible for the distinguishment and the difference between the self and the non-self peptides. As HLA are highly polymorphic glycoprotein molecules that can present self and non-self peptide to the immune system. The immune system may produce antibodies against the non-self HLA, and this could be due to the sensitization to previous transplants, pregnancy, or blood transfusion. Any reason there could be. Anti-HLA antibodies directed against non-self HLA will bind to the target and causes a hyperacute or the acute rejection. Here I would love to state that the T cells represents the class 1 HLA that correlates with the hyperacute or the acute rejection, and the B cell represents the class 1 as well as the class 2 HLA that correlates with the chronic rejection, the chronic graft rejection and the graft dysfunction. The antigen-antibody reaction leads to the cell lysis examined under inverted phase contrast microscope with the help of Eosin dye. The dead cells observed as black spots while live cells appear bright. Here in the picture you can see there is a potential recipient and the serum of the recipient. There might be the presence of antibodies that are also known as DSAs, and we need to detect them. If there is the presence of these antibodies, they will go and bind to the donor lymphocytes, and when they will bind to the donor lymphocytes, they will activate the complement cascade, and eventually it will lead to the destruction of the cells and the graft will get rejected. That is why we need to detect these DSAs, and we need to access this CDC test to detect the cytotoxic antibodies in the recipient serum. There are two types of CDC crossmatch, namely auto crossmatch, that is also known as the autologous crossmatch. Herein, the patient cell are tested with the patient serum. That means the test is being done within the same individual. And the next is the allo crossmatch, that is allogenic crossmatch, that is done with the another individual but of the same species. Here, the donor cell are tested with the patient serum. Here in the image you can see the allo crossmatch. In the allo crossmatch, donor cells are tested with the patient serum, and in the auto crossmatch, the patient cells are tested with its own lymphocytes. Here comes the reagents used in the CDC crossmatch. For the negative control, that is also known as NC, it confirms the absence of non-specific cytotoxicity. In the NC, for the negative control, we don't have any antibodies present in that, and it ensures test validity by showing no unintended cell death. Next is the PC. For the positive control, it contains the known antibodies, confirms that complement and assay conditions are functioning properly by inducing the known cell lysis. Here comes the DTT, dithiothiotol. It is a chemical that inactivates the IgM antibodies to distinguish clinically relevant IgG-mediated cytotoxicity. Here, let's have a look on the DTT mechanism. As you can see, there is a IgM pentamer. IgM is a pentamer. It is a vast structure that highly depends on its disulfide bond. Here, the disulfide bonds hold its structure intact and helps the IgM pentamer to work efficiently. But we do not need this IgM because it does not correlate significantly with our clinical preferences, as IgM is naturally occurring and it is an autoantibody. That is, it is an autoantibody. That is why we need to rule out this antibody, and we specifically detect the IgG, which is responsible for the graft rejection. That is why we are using DTT here. Let's have a look on the mechanism of DTT, that how it breaks the disulfide bonds. Firstly, DTT will activate itself. It will target the disulfide bonds of the IgM pentamer. It will break the protein and get it split. And after splitting it, it stabilizes itself by forming the internal bonds within itself. This is how DTT works. It will deactivate the IgM pentamer, as when the disulfide bonds of the IgM pentamer will break, its activity will be inactivated and it can't work properly. On the DTT mechanism, here you can see there is a structure of IgM antibody. IgM antibody is basically a pentamer. It is a vast structure and it is highly dependent on its disulfide bonds. These disulfide bonds help the structure to remain intact. DTT acts upon these disulfide bonds. Let's have a look how DTT breaks the disulfide bonds of the IgM antibody. When the DTT acts, DTT firstly activates itself. DTT will attack on the disulfide bond. DTT will break the bond and it stabilizes itself by forming the internal bonds. Here, this helps in the ruling out of IgM antibody, and we can select, and we are selectively going to detect the IgG antibody. But why are we ruling out the IgM? We are ruling out the IgM antibody as it is a naturally occurring antibody, or it is also the autoantibody, but it can strongly activate the complement that can cause the false results. That is why we are ruling out the IgM antibody, and this allows us the specific detection of DSAs in the test. Next we have is AHG, that is anti-human globulin serum. It enhances the detection of weak IgG antibodies by amplifying the complement-mediated cytotoxic reactions, as it detects those antibodies which are weakly bind, and those are the IgG antibodies. As this is AHG, that is anti-IgG. It goes and binds to the IgG, that creates a more big immune complex that helps for the easier detection of the weak IgG antibodies, and it amplifies the reaction. Here the AHG increases the sensitivity of the test, and by depleting the disulfide bonds of IgM by DTT, it increases the specificity of our test. Next is the rabbit complement. Rabbit complement is the source of complement proteins. It activates the complement cascade, leading to the cell lysis when antibodies bind to the donor cells. Histopac is a density gradient medium used to isolate the lymphocytes from the whole blood. McCoy's 5A medium. Here we are using the McCoy's 5A medium and not any other media, because what we need is the stable lymphocytes, and it maintains the viability and physiological conditions of the lymphocytes during the assay preparation. We do not need any proliferation of the cells. That is why we are using here the McCoy's 5A medium, and it prevents the false positive and the false negative in the test. Next is the paraffin oil. Paraffin oil prevents the evaporation of micro droplets during incubation in the microtiter plates. Eosin dye basically works on the vital dye exclusion principle. Vital dye used to identify the dead cells, while the dead cells absorb dye, indicating the cytotoxicity. It basically works on the membrane integrity. Rosette T and B cell reagents. Here we are using the rosette T and B cells to separate and enrich the T and B lymphocytes from the donor. Here is the workflow of our test. That is, we'll start from the isolation of donor lymphocytes. We'll incubate with recipient serum. We'll proceed with washing, complement addition, and an incubation. After that, we'll stain with eosin dye. We'll observe under the inverted phase contrast microscope, and at last we'll score and interpret. Moving on to the key requirements for the CDC crossmatch. Firstly, the blood sample should be collected in the ACD vials for the CDC crossmatch. Here, ACD stands for acid citrate dextrose. Here, the citric acid stabilizes the pH of the blood sample, and the citrate is an anticoagulant, whereas the dextrose is the source of energy for the cells to keep them alive. As because in CDC crossmatch we uses the live cell, and here the ACD vial supports the viability of the cell. Next we have is recipient serum, donor lymphocytes, DTT, that is dithiothiotol, rosette T and B cells, McCoy's 5A medium, paraffin oil, negative control, positive control, histopac, complement, AHG, eosin dye, falcons, Terasaki tray, microcentrifuge tubes, and Pasteur pipettes. Here is the initial step of the test, that is the preparation of McCoy's 5A medium. Here we will dissolve the 0.5 grams of McCoy's powder in 50 ml sterile water. We'll adjust the pH to 7.4 using NaOH dropwise. We'll filter the prepared media 2 times with the millipore filter to avoid any kind of contamination in our test. As CDC is known as the mini transplant, we cannot bear any chance of contamination there. Let's start with the arrangement and labeling of respective falcons. We will label the falcon tubes as PT for the patient T cells, PB for the patient B cells, DT for the donor T cells, and DB for the donor B cells. Here We will add the samples, we'll transfer the blood sample from the ACD vials to the patient tubes, as the patient blood will be transferred to the PT and PB falcons and donor blood to the DT and DB falcons respectively. We'll add 2.5 ml of McCoy's media to each falcon, and after that we'll add 100 microliter of rosetting reagents, that are T and B cell reagents, to the each falcon. We'll mix properly by inverting the falcon slowly and incubate it for the 20 minutes at room temperature. Here you can see in the picture the arrangement and the labeling of respected falcons for the three donors respectively. Next we'll move with lymphocyte separation. In fresh falcons, we'll add 3.5 ml of histopack. We'll carefully layer the sample over the histopack. Here in the picture you can see the layering of sample over the histopack, as histopack is the density gradient medium. We'll centrifuge it at 2000 rpm for 20 minutes. After centrifugation, we'll collect the white cloudy buffy coat layer, that is of lymphocytes. We'll transfer the lymphocytes to the new labeled falcons. We'll add 1.5 ml of media. We'll mix it gently. We'll centrifuge it again for 2500 for 10 minutes, as our cells are fragile. After centrifugation, we'll discard the supernatant and we'll resuspend the cell properly. Here in the image you can see, at the left side, the first image, you can see the blood components are separated after centrifugation. Here the middle layer is the white cloudy buffy coat layer. The upper layer is a supernatant. We have to discard it. Basically it is the plasma constituents. And at the bottom of the falcon you can see the erythrocytes. These are arranged to their densities just because of the histopack media, that is the density gradient media. After that, after collecting the buffy coat layer from this falcon, we'll transfer it to a new falcon and we'll add McCoy's media to it. After centrifuging for 2500 rpm for 10 minutes, we'll get the cell pellet and we'll disturb the cells properly and resuspend the cells properly in the falcon. Here is the plate setup. It's all about the tray that is known as the Terasaki tray. It comprises of the 60 mini wells or the micro wells. At the column 10, at the very left of the tray, you can see the column number 10 that represents NC, negative control. At the right side, you can see the first column, that is the PC of positive control. Column number 2 is the DTT, that is used to control to break the disulfide bonds of IgM pentamer. At the third column, you can see the 1:2 DTT treated serum. This is a titered column where we'll check the strength of the antibodies. And in the column number 4, that is star, that represents the non-complement. Here are the rows, A to F rows for the PT, PB, DT and DB. Here in the next image, you can see it properly here. 1 to 10 columns are presented at the topmost of the tray. Here you can see the A to C rows are denoted for the donor T cells and donor B cells. And for the patient respectively, D to F rows for the patient T cells and patient B cells. Here what we can observe is there are 2 rows of T and 1 row of B. Why? Why there are 2 rows of B and 1 row of B? Because there is a phenomena that is working there and we need to rule out that. It is known as the prozone effect. Here comes the prozone effect, an effect where the antibodies are present in excess. The phenomena where excessively high concentration of antibodies are present. Due to high concentration of these antibodies, there will be more antibodies and the HLA epitopes are less. So there will be the competitive binding, and because of this competitive binding, if the cross-linking is not done properly there, the C1q protein will not bind properly and can't activate the complement cascade. If the complement cascade is not activated, there will be no MAC formation and there will be no cell lysis. And it disturbs the proper antigen-antibody lattice formation and reduces the effective cross-linking and complement activation. will lead to false negative and weakly positive results even when clinically significant antibodies are present. But why does it happen? Too many antibodies saturate antigen sites on the lymphocytes. Antibodies cannot cross-link efficiently. Complement activations become inefficient and the result will be reduced or absent cell lysis that gives us the false negative result. Here in the picture you can see if there are too many antibodies present, many antibodies present, there will be the competitive binding between them. No effective cross-linking will be there and the C1q binding will not take place. Due to this, the complement cascade will not get activated, the reduced complement activation, and eventually it will lead to no cell lysis. That is why we need to rule out this prozone effect. Here we'll move with reagent addition and cell loading. Reagent addition to the plate. We'll add 5 microliter of paraffin oil to each well. We'll add 2 microliter of NC, PC, DTT in the respective columns. Seed will prepare the serum dilutions. Prepare 1:2 dilution for the 50 microliter of patient serum plus 50 microliter of media. We'll add only to the 1:2 column. We will do not add the serum to the NC and PC columns because NC and PCs are the serums itself. We'll add neat patient serum to the first T and B rows. For the cell loading, we'll add cell as follows: DT, A and B rows, DB for the C row, PT for the D and E rows, and PB for the F rows. We'll incubate the plate at room temperature for 45 minutes. After the incubation of 45 minutes, we'll proceed with washing. We'll add 10 microliter of media to the each well. We'll centrifuge at 800 rpm for 1 minute and 30 seconds. We'll repeat the washing 3 times. As we are repeating the washing steps for the 3 times, this will increase the sensitivity of our test. Next is the AHG and the complement preparation. Here we will prepare the AHG and the complement. We'll take the 3 microcentrifuge tubes. Tube A. Tube A will contain the 150 microliter media plus 1 microliter of AHG. In the tube B, we'll take 250 microliter of complement plus 87 microliter of media. This makes a diluted complement. We will transfer 37.5 microliter of AHG mixture from tube A to tube B, and after that we'll discard the tube A. And in the tube C, we will take 200 microliter of neat complement. Here, we will add the complement addition to the plate. In the star column, 5 microliter of no complement will be added, that is the McCoy's media. In the C and F rows, we will add 5 microliter of neat complement, that is of the B rows. As B rows, B cells are more fragile and have more HLA density as compared to the T cells. That is why they are more fragile and sensitive. So that we will add the neat complement to the C and F rows. In the A, B, D and E, there will be the addition of 5 microliter of diluted complement because we are adding AHG to it. That will increase the sensitivity of our test. It will detect the weak IgG antibodies in the test and will make easier to detect those weak immune reactions. It will amplify the reaction and so that we can easily detect them. As AHG is inherently toxic, that is why we will always use AHG in the diluted form only. After that, we'll incubate for 1 hour and 30 minutes. Then we will add the complement to the plate. This is a pathway that takes place. But why only classical pathway for the complement activation? Because here the antigen and antibody complex is taking place. That is why it is termed as the classical pathway of complement activation. When the antigen and antibody will bind together, the Fc portion of the antibody will get exposed and this is the only trigger. If there will be two adjacent antibodies are not close enough, then there will be no C1q binding take place and the complement will not get activated. So for the activation of the C1q binding, the two adjacent antibodies must be close together so that the cross-linking can take place and the C1q can activate the complement cascade. When the C1 complex takes place, it activates the C2 and C4. Here, the C2a and C4b fragments will activate the C3 convertase. C3 convertase will activate the C3. Here, the C3 hydrolysis will take place. C3b and C3a will get in fragments. C3b will cleave the C5 into C5a and C5b. From the C5b to C9, they will together form the MAC. MAC stands for the membrane attack complex that will punch the hole in the cell membrane. Therefore, the fluid will rush in, the cell will swell and eventually it get burst. This is how the cell lysis takes place with the help of complement proteins. After the incubation of 1 hour and 30 minutes, we will add the 10 microliter of Eosin dye to the each well. As Eosin is an acidic vital dye, it works on the vital dye exclusion principle. It cannot enter the living cells as it works on the membrane integrity. It enters only the cells with the damaged membranes. When the cell membrane integrity is loosened, is compromised, after that only the dye can enter the damaged membrane. The live cells will take up the dye and the live cells would not take up the dye and will appear as bright, and the lysed cells will appear as the black spots. Here in the image at the left side you can see there is extensive cell lysis which indicates the complement mediated cytotoxicity. You can observe the black spots here, thousands of black spots are there. That means the complement mediated cytotoxicity happened here. And on the right side of the image you can see there are many bright cells, bright and intact cells, which shows the absence of the cytotoxicity. Here is all about the result reading. Trays are read with the inverted phase contrast microscope. Dead cells will take up the eosin and will appear as black spots. Live cells will exclude the dye and will appear as bright. Here is the quality control criteria in our CDC test. For the controls, that is NC, for the negative control there will be no lysis. This is what are the expected results because it does not contain any known antibody and it confirms the viable cells. For the PC that contains the known antibodies, it should give the results of high cell lysis that confirms the complement activity. And for the auto control, it should be negative and it rules out the auto antibodies. If there is a presence of auto antibodies in the patient, there might be any autoimmune disease that is there. Here is the illustration of the positive and negative CDC crossmatch. For the negative CDC crossmatch, you can see there is no donor-specific HLA antibodies in the recipient serum. That means no antibody binding. That is why it is a negative crossmatch. There will be no cell lysis, no antibody binding. That means there will be no C1q binding, no C1q binding. It indicates there will be no activation of the complement cascade. That means no MAC, no membrane attack complex, and there will be no cell lysis. That is why it is a negative crossmatch. And for the positive crossmatch, donor-specific HLA antibodies in the recipient serum. Antibodies will bind and will activate the complement. The MAC will form and the MAC will eventually lyse the cells. This is the positive crossmatch. Here is the scoring for the CDC crossmatch. CDC score number 1, that is 0 to 10% cell death. The reaction strength is negative here. There is no significant cytotoxicity and the donor specific antibodies are there is no DSAs detected. That is why the cross match is acceptable. For the CDC score number 2, there is 11 to 20% of cell death. That means the reaction strength is negative. There could be the background cell death and it is considered as non-reactive. For the CDC score number 4, there is 21 to 50% cell death. it is doubtful and weakly positive, but as it sounds positive, we need to worry. There is possible low level of antibodies, but we need the required repeat testing or confirmation. Here for the CDC score number 6, there is 51 to 80% of cell death. That indicates the reaction strength is positive, and there is moderate cytotoxicity. There is the presence of DSAs, and there is increased risk of rejection. For the CDC score number 8, there is 81 to 100% of cell death, that is strongly and highly positive. There is high cytotoxicity and strong antibody mediated incompatibility. We cannot proceed for the transplant here. T cell versus B cell CDC interpretation. For the T cell CDC positive, there is class 1 HLA antibodies and there is high risk major contraindication. For the B cell CDC positive, there is class 1 or class 2 antibodies present, suggests class 2 sensitization and will interpret with caution. Here are the limitations of the CDC test. As CDC test detects only the complement binding antibodies, it cannot detect any other antibodies. It only detects the cytotoxic antibodies present there. Next is low sensitivity. It has the low sensitivity. It might miss the weak antibodies. That is why we are using AHG, the extended incubation periods, and we are increasing the washing steps to increase the sensitivity of the test. But because it has low sensitivity, we'll go for another immunological pre-transplant testings, that is flow crossmatch, Luminex crossmatch testings, antibody screening. It cannot identify the antibody specificity. It cannot specifically identify that antibody is directed to which antigen. That is why we need to go for some other test also to get the specific antibody specificity, for example SAB. It cannot quantify the antibody strength. It cannot quantify the antibody strength, as it can identify the antibody but not its strength. So to detect its strength, we'll go for the Luminex crossmatch. That will give us the MFI, that is mean fluorescence intensity, that will help us to quantify the antibody strength. It cannot detect the memory or the historical sensitization. Because it cannot detect, that is why we are using the antibody screening here to detect memory or the historical sensitization. So it was all about the limitations of the CDC crossmatch. Thank you and have a nice day.